just so everyone knows, this will be recorded for hopefully future posting of it for others who can't be here today. I welcome everybody who has joined us and who is still joining us. I'm going to tell you today about the Blueprint Neurotherapeutics Network, which has a receipt date coming up in August. And at the end, hopefully there'll be plenty of time for me to answer questions. Tim, you have any logistics?
I just want to give everyone a heads up how they're going to ask questions. It's going to be in the Q&A box. The way that you find it, if you're looking at your laptops or computers, if you hover over the center of the screen, you'll see 3 dots. If you click on that, you'll see the Q&A where you can click on Q&A. And that is where you're going to ask your questions. Try not to use the chat function. Please use the Q&A field. And all the slides will be emailed to everyone that has registered for this webinar.
All right. So, thanks again. Get started. So, the program vision for the Blueprint Neurotherapeutics is to combine the strength of NIH with industry expertise and NIH investigator-initiated ideas for small molecule drug discovery. You need to have a small molecule, and you need to have a strong disease assays and models in place at a minimum to come into the Blueprint Neurotherapeutics Network. We can provide everything else to you and wrap around you, but the disease biology, the small molecule starting point, really the responsibility of the investigator. What we do is team you up with advisors with extensive pharma experience, give you access to our industry standard contract services to create the whole network for you. The program goals are to de-risk the project to the point where industry will invest in it, and hopefully allowing for potential new drugs to reach patients efficiently. We need someone who's going to be able to take this to phase 2 and beyond, involved hopefully by the end of the project, in order to keep a seamless transition going. We look and seek to identify the best ideas for translation of NIH research communities research through this funding opportunity and the associated infrastructure. We provide the funding via grant funding and access to the necessary resources, typically being lacking in our research community. And one of the main features of this program, which is really different than a lot of the programs you'll see elsewhere in NIH, is we've taken great care to preserve the PI and institutions' intellectual properties to facilitate licensing. And that has really paid dividends for us in terms of our programs being successful and having projects move forward. So the program is open to both academics and small businesses. The grant funding is, as I said, focused on small molecules, not on biologics, not on conjugates, not on large peptides, really small molecules. We'll consider peptides up to 5 amino acids on a typical thing. If you have a question about a peptide, we can certainly talk about it. And I've already mentioned a number of times, you're really accessing our consultants and CROs. If you don't need our expertise for the consultants or the contracts, this is not going to be the right mechanism for you, and you should talk to me about what might be a better mechanism for you. Projects can enter at the discovery phase, meaning I still have medicinal chemistry left to do. You have to come in with at least a well-validated hit compound that you're going to optimize through a medicinal chemistry effort. The development phase is, I'm really done with medicinal chemistry. I have my candidate, or I'm down to a couple compounds. One of them is going to be my candidate. That's a development project. You're really trying to get into the IND-enabling toxicology as quickly as possible to move to the clinic. This program is open to projects within the mission of the 9 participating institutes and centers. As listed on this slide, you can see NINDS is where my home is, but we also have aging, alcohol abuse and alcoholism, NIDA for drug abuse, the Center for Complementary and Integrative Health, mental health, child health, dental, and the Eye Institute all have representation in this program, and we've had projects that span, I believe, all of these institutes at one point or another over the course of the last 7 years I've been doing this program. So the infrastructure I talked about is in place in a customized fashion, but what's paramount to all of our programs is we create a lead discovery and lead development team around the program that consists of the principal investigator and our consultants. We always have a lead consultant and the principal investigators co-chairing this team. This team meets every 2 weeks. It's very interactive. Cards are on the table as we move through the project. It also has NIH staff, the program staff, as well as science officers with individual ICs that are supporting the program, whose mission you're in, can be there for a resource as well. You are, as I said, through the NIH grant, responsible for, at a minimum, of the bioactivity efficacy studies, things related to disease biology. That would be to be through your institution in your collaboration in the grant budget, meaning either through your own institution or through collaborators or contracts on your side. In addition to that, we have access to contracts from NIH. They include medicinal chemistry, they include PK toxicology, drug manufacturing formulation, and clinical trials. But I should point out the medicinal chemistry contract can be synthesis, it includes in vitro ADME work, it also includes computational chemistry. It can be any or all of those components together, depending on what your program needs. You can do all of this stuff yourself, in theory, but if you're doing all this and you have it all within your budget, you have to do it within the limits, and I'll talk about those limits later. But it may be better to consider other mechanisms if you don't actually need any of our help on this, because we may actually slow you down. This is a very interactive, milestone-driven program. If you have your whole system in place, this may or may not be the right program for you. I really encourage you, when you have questions, to contact us. The important thing is we do make it customized to whatever you need. So if you are doing medicinal chemistry, you might need just in vitro ADME to support what you're doing. Or you might have a PK core that can do all your mouse and rat PK, but you don't have the ability to do large animal PK or the toxicology stuff that's coming down the road. We can customize what's being done in your lab and what's being done through our contracts to meet your program needs. So how do we advance the projects? We advance them, depending on their entry point, through a set of milestones. And anyone interested in, I'll touch on this in a moment, what our milestones are. There's sort of a standard set of BPM milestones depending on your entry, and then there's customized milestones depending on the disease biology that may be important for your program. But we seek to enter projects in with strong science within our mission and exit them to external funding or other grant mechanisms, or through, unfortunately, through attrition if we hit scientific roadblocks that make it impossible to move the project forward and missing milestones. I've already covered this slide where, the flexibility of the contract resources and what you do. But the purpose I put this in is it's really important as part of your application to clearly delineate what it is you're looking for the BPN to provide you, as well as what you're going to provide. And to that end, there's some requests I'll hit on again later about having a table that clearly shows what that is, having a screening funnel show you what your throughput in, having IP agreements, and all that. What I failed to point out, and I really should point out, I'm going to go back 2 slides just really quickly. There are 2 key places where you're really going to generate intellectual property: the medicinal chemistry contract and the drug manufacturing formulation contract. Both of those contracts have special clauses associated with those contracts that assign the intellectual property to your institution. They have what's called a termination of exceptional circumstance clauses. It basically says that any chemistry to do with your compound or analogs thereof belongs to your institution. If, For instance, the MedChem contract was to create a catalyst that was used generally utility. They could patent that themselves, but they would have to give you a fully paid up worldwide license that's transferable. So again, it's preserving your intellectual property and your ability to prosecute your project. The same thing is true for the drug manufacturing formulation contract. It also has a determination of exceptional circumstances. It's a two-phase grant mechanism. Everybody comes into the preparatory phase no matter what. So I get that question a lot, and there is no going around that. Everyone's going to enter in at phase 1 of U44 or UG3 phase, one of those 2 points. What happens in the preparatory phase is dependent on where you're entering the project. If you're entering at the development phase, it might be that you need to be repeating an efficacy study or completing the dose range finding tox studies and stuff to get to your go, no-go starting point. But if you're coming in at the earliest stage of exploratory, it's going to be all about show me the assays up and running, that we have an assay that has the throughput to run an SAR program that's performing, the metrics on it are good. Benchmark your starting compound. That, I see those a lot of times in review because it's not clearly stated in the application, but it's important to say, hey, this is only an early hit. I've got this great set of preliminary data. This is why I believe this target or this pathway is great. I've got this hit, and I have this information about compounds like this that hit this target, but I don't have the information on this compound. That's going to be what the preparatory phase is all about. It's about getting benchmarking data for that compound, and that's what we can do in phase 1 or in the UG3 phase. So that's really where the first phase is. It must have a go, no-go milestone associated with it in order to move forward to the next phase. It is a administrative review to go from UG3 to UH3 or phase 1, U44 phase 1 to phase 2. It's an internal meeting. It's not a new application. It also means that all years of your grant budget must be there, not just the preparatory phase. All years of the grant budgets must be part of the application process. To that end, projects can enter, as I said, any stage, but there are, and you can come in SBIR or non-SBIR version, but if you come in on the non-SBIR version, it's important to note that if you exceed $500,000 in direct costs in any year of the grant, including the UH3 phase, you need to be contacting me 8 weeks prior, meaning like right away, that you want to, if you're coming for the August date, that you want to do that. Most people do not exceed that because they're utilizing our contracts, but in cases where you're doing your own medicinal chemistry or something like that, you may well do that. You need to be ready to go when they hit the ground running when the contract or when the grant is awarded, meaning you shouldn't have, I want to hire chemists. This is a milestone-driven program, and you can have your baseline chemists that are there working on it, your baseline biologists, but if you're going to need to hire a bunch of people, keep in mind that you could be firing a bunch of people if you miss the milestone. So you don't want to put yourself in a position where this is the only source of funding for those people, and that you plan ahead for both success and failure. Not all projects come in at development phase for all ICs, meaning, for instance, NIMH and NIDA will not accept programs at the development phase under the Blueprint Therapeutics. NIDA has their own FOAs, funding opportunity announcements, for development projects that they would prefer you use. NIMH wants to know more about the projects. They don't want to take something with that level of expenditure right out of the gate. If you're coming under the SBIR, there's the difference in these budgets is the $300K in direct cost versus $500K in total cost, $1.5 million in direct cost versus $1.5 million in total cost. So there's a little difference there in what you can possibly max out at in the U44, but you don't have to ask permission on the U44 for your budget. So that's a little different there. The milestones, I promised, my progression is sort of listed here. These are my generic milestones I have for the program. We customize them to each program, but if you're coming into the exploratory stage, as I mentioned, it's about showing me your assays working, show me you have a good target product profile in the funnel, screening funnel, show me you have chemistry that's robust and ready to move into a lead optimization program, either hit to lead or lead optimization. At the end of that period, with a good solid data package for a solid data-driven plan to be in place. That's what we're trying to produce. Once we get to hit to lead, it's about advancing the SAR. It's about showing me in vivo pharmacology by any route of administration. By the end of lead optimization, we're picking our candidate. We're picking our candidate by showing by the intended route of administration according to our target product profile that we have good PD pharmacodynamics and we have good therapeutic index by running, for instance, the dose range finding in the rodent species and showing me a window. The pre-development activities are things like large animal dose range finding, salt screen polymorph work, showing me we actually have a synthetic route that we can scale material up in a cost-effective way. We do not have unlimited funds, and I can't do a 25-step synthesis with a 2% yield that costs millions of dollars. So I won't be able to do something like that. So we have to fix that, or that could be a reason not to proceed. IND enabling studies are typically done through our contracts and are done to industry standard GLP studies. GMP synthesis, again, we can do it through our manufacturing or through something you've worked out, especially if that happens to be a peptide or something. It might be some beneficial for you to go through your own clinical trial material, same thing. And then lastly, where you typically end the program within Blueprint at the first in human experience, single ascending dose study. So we open the IND, have those discussions, and move the project forward. The maximum grant duration is 5 years for the combination of the UH3 and UG3, UH3, or for that matter for the U44 phase 1-2, depending how you laced out the phase 1. You can plan on the intellectual property being determined by U.S. patent law. So inventorship, if our contractors, our medicinal chemistry contractors, or drug manufacturing pharma contractors work on and have an invention, they'll have to be an inventor on your patent. That's just patent law. But as I said, the Blueprint itself is not trying to seek any intellectual property rights. They're trying to make sure that those contractors are assigning the intellectual property rights to you. So prior to grant award, we will make sure you have upfront IP agreements in place with all potential inventors. These must address things like who's going to hold the IP, who's going, if you have multiple groups involved, who's going to take the lead on that. Some agreement in place. Our consultants will sign IP agreements. As I said, the contractors already have special clauses in there so that it's contractually determined there. But the whole purpose is that unencumbered IP. There's a lot of information on our website, and I want to point this website out to you all. What you can find here is listed down amongst this at the bottom is all the staff, and all those are active links if you need to email us. On the left, you'll find the funding opportunities on the left-hand link under VPN funding opportunities. You can get an overview slide of the program in a poster that's listed right there at the bottom of the second paragraph. And you can get what our resources are, who our external oversight committee is, who our consultants are. The important thing is you do not need to budget our consultants. You do not need to budget our contractors into your budgets. You just need to make a table that says that's what you're going to use. If you don't have what you need, I know I saw some questions, there's a program called Ignite, for at least for things within NINDS's mission, that can get you what you need. Those include things like assay development, so develop an assay and run a screen under PAR 18762, or develop a compound, do some preliminary SAR, and run an in vivo efficacy study so I can get the preliminary data to be ready for a Blueprint program. Or if you're developing and validating a model system, if you're trying to do biomarkers and so forth like that, again, you can do that within these. Those budgets are typically $250,000 a year, up to $750,000 across the 3 years. Are also milestone driven. They're staged mechanisms. Rebecca Roof is your contact for that. I'm going to just skip that slide and move ahead here. And the important thing I want to cover here is some things you really need to do. and I can't tell you how often I get asked questions about what to do. And at the end of the day, I have to open the funding opportunity announcement myself and make sure I'm giving you exactly what's in there, because it's going to be prosecuted exactly the way it's written in the funding opportunity announcement. So you read those carefully. Discuss your proposal with the NIH staff, myself and others, and the Home Institute. If you're not with NINDS, make sure you have a conversation with whatever institute your disease is in, because I'm going to get money from them, and that home science officer is going to be instrumental in me, if you score well, getting your application funded. Contact the SBIR program directors if you have SBIR-specific issues. Stick to the page limits that are required. Going over them, stuffing your application will end up getting your application withdrawn, and that happens before I ever even see it. Put forth a solid scientific premise and address rigor in your research strategy section. Don't bury it in the vertebrate animal section. It needs to be as part of the discussion of the preliminary data. Was it done in a rigorous fashion? Was it blinded? Was it properly powered? All of those good things. Address the obvious criticisms that you might have. If your compound's got a flag in it, come right out and say, I have a flag in it, and that's going to be something that we're going to try to address, and here's how we're going to address it. Because the reviewers will assume if you don't say that, you don't appreciate it. And oftentimes that's not the case. Sometimes people think it's really obvious they're going to do that, but from a review point of view, it's better to address the obvious, even if it's just a sentence. Say, yes, I clearly understand that. Clearly indicate what's going to be done as part of the grant and what you expect the BPN contractors to do. I recommend a table strongly that clearly lays out, here are the assays we plan to do, here's the throughput of those assays, who's going to be responsible for running those assays, how many times we're going to run those assays in a given year. Include budget sheets for all years. I mentioned that again. Every receipt round, I try to contact people before if they submit early to get me their revised budgets quickly, because they've left out year budgets. As I said, scientific premise is really critical. Explicitly discuss it. Even the prior publications discuss in a detailed manner why this data is good data and that you can rely on it. Complete all your registrations 6 to 8 weeks in advance. So if you want to come to the August date, now is the time to get yourself all your registrations. Make sure you have all ERAs, DUNS numbers, everything that's required. I know DUNS numbers are actually going to get phased out at some point and changed over to a new set of numbers. I'm not sure exactly the date that's going to happen, but I know that that's something that you need to keep an eye on and make sure everything from a registration point of view is up to date. So consider submitting your application earlier. I do try to look at applications a couple days before the due date. If they're in, really quickly for obvious flaws like the budget sheets didn't come through or something's missing, or you've got something extra in there that needs to get removed. If you're applying for the UG3, UH3 and any year exceeds $500K in direct costs, you need to submit ARA, which means I need a pretty detailed budget. Actually, I need a completely detailed budget with justification for why you're going over $500,000. Consider using our contract, and why, if you're not using our contracts, justify why not. In that scenario, if you're going to exceed it, because I'm going to have to justify why not. Talk to your tech transfer and business development groups early. You need to plan for funding for patents and licensing activities for which you cannot put in your grant. Some things not to do. As I mentioned, don't plan on this to be your sole funding for your lab. It's a milestone-driven program and can and has in the past ended abruptly due to science, and in at least one case and due to competitive information that came in. Do not under-resource your budget to avoid the limits. It's important to put in what you think and put the ARA if you really need it. Please do not plan for BPN contracts for disease biology, meaning I can't run your primary assay, your SAR driving assay. The contractors I have don't do that. They just have too many diseases and too many different kinds of assays. Don't plan on one-off in vitro ADME experiments either. So if you're coming as a development project and you just need one or two things to fill your hole that you're missing, put that in your budget, because it's far more efficient for you to do that than it is for me to put a whole big contract in place to do a couple of things. Please do not code your application as a clinical trial if, in fact, you're not going to run the clinical trial yourself. If you plan to use the BPN contractors, then this is not a clinical trial application. I handle human subject stuff through the contract process, not through this grant side. But if you do plan on doing a clinical trial and you plan on doing it yourself, you're probably going to have a delayed onset. So make sure you say so. Some things to really include: make sure you have a target product profile. And there's some links to do that at our website. Make sure you have a screening funnel. Either include a pictorial screening funnel or a table or something. And for PIR 18546, which is the UG3, UH3 mechanism, applications must include an intellectual property strategy in the other attachments. So you need to have that. That is the only other attachment you can have. Nothing else is called for. Don't put anything else under other attachments, just that. If you are coming under the SBIR version, then the IP strategy is part of the required commercialization plan, and it'll be covered there. As I mentioned, the target product profile, this is, you can find it on our website, some FDA guidance on what sort of one looks like. If you're not familiar with one, it typically has a minimum acceptable and an ideal. We're really trying to tell us what our patient population and it's going to look like, what kind of delivery mode we're going to have, whether it's going to be oral, IV, how often we're going to give it, ideally and acceptably, and what kind of efficacy we expect to see, what kind of side effects we expect to see. It's all about planning with the patient in mind, and that really dictates what animal models we should have in place, potentially what toxicology we need to be running, and so forth. I'll give you an example of a screening funnel that I presented from the Petrukin project to give you an idea of what a pictorial one looks like versus a table. But here you can see it has all the components. It says I'm going to start with this many analogs. I'm going to run it through a primary assay, a secondary assay. My advancement criteria is there. Here's what I'm expecting for a selectivity ratio. I'm going to go into my in vitro ADME. I'm going to go into my first tier, my second tier. I'm going to have eventually PK and broad panel screening stuff on other down the road. And who's responsible for doing that? Whether it's BPN or through the AMRI contract or whether it's the home lab in this particular case, that's all color-coded. But you could also do this on just a simple table. I've seen both be very effective in applications. Key notices, I point this out as I mentioned just a minute ago. Do not put stuff in that's not called for. If you put extra things under other attachments or put it in like journal articles under supporting documents, letters of support, your application will end up being withdrawn by CSR before it ever gets reviewed. They'll just say it's not compliant and withdraw the application and send it back to you. So follow the FOA carefully, I implore you. There's very little I can do. Once they do this, it's pretty much a done deal. And if you haven't submitted it early and there's no time to resubmit, you're just out of luck until the next receipt date. So speaking of the next receipt date, that is August 9th, and our next one after that is February 11th in 2020. They're reviewed by a special emphasis panel internal to NINDS that is consists of experts in drug discovery, experts in disease biology, experts in general in the area of neuroscience. Both the SBIR and the non-SBIR applications are reviewed by the same panel at the same time. The following indications are all listed in the FOA, and you can look for what each IC is interested in doing. And the contacts are also listed in the funding opportunity announcement. If you have questions for disease area biology and that institute's interest in what you're planning to do, that's a good stop along the way. So with that, I'm ready for questions. So Tim will give me questions, and I will try to answer them in order. You can just keep plugging them into the Q&A, and I will take them one by one I apologize if I lose my voice part way. Is it acceptable to have a project between the collaboration between my lab and that from a foreign country? The answer to that question is depending on which funding opportunity announcement you're discussing. So yes, it is possible to have a foreign component, and you can even have a foreign application under the PIR 18-546, which is a UG3 mechanism. If you're coming under the SBIR mechanism, you must follow all the SBIR rules, which generally does not allow any of the funding to go to foreign components or foreign institutions at all, or foreign CROs. So you look to see which application coming in. In fact, sometimes that's why a small business may choose to come into the UG3 mechanism because of established relationships they have. This funding is aimed more towards the preclinical testing, and is this funding more preclinical testing or for human trials of some sort? It is really all preclinical to get you to the first. It ends with first-in-human trials. So if you already have a compound, in fact, anything directed beyond phase 1 is out of scope. Anything directed just at phase 1 is out of scope. So if you're just coming in to do a clinical trial, then this is not the right mechanism for you. We can talk about there's experimental clinical trials, FOA, and NINDS, for instance, that you could go to. How many awards are issued per year? I get asked this question pretty much every time. It is totally dependent on the number of applications I receive each year and how they score. I typically launch somewhere between 2 and 4 projects per year for the 2 different receipt dates each year, so about 2 projects each time. The success rate is very similar to what we see for any other grant application. Will BPN support target identification of a compound already in clinical development? No, it will not. That is sort of an R01 kind of activity, and I would recommend you go there. Who decides the subcontractors? Can we bring someone in and they will be the BPN network? So the subcontractors, if you're using our contractors, BPN, then if there's 2 listed there, it'll be bid between the 2 of them. And whoever wins the bid will get it. But for like MedChem, there's only 1 contractor, that's AMRI. If you bring in your own contractors and you have them in your budget, they're not part of the network per se, but they're part of the project team. And so by virtue of that, they're going to be a BPN contributor, but that would be totally on your side and be part of your budget. So if you want your own contractor, specific contractor, then you need to include that in your budget, get the letter of support, get the, the quotes and everything so you know what to budget and all that. What is the max budget, and does that number include BPN contractors? Absolutely. It does not include our budget or on the contractor side. The budget should reflect what you actually need. What is the split between company, like typical SBR? I assume you're asking me about the requirements for SBIR there, and they're the same as any other SBIR program, which means in phase 1, 51% must stay with your company. If you're in phase 2, said that wrong. 66% must stay with your company in phase 1, 51% in phase 2. If you have a fee-for-service contractor that SBIR the group determines is actually fee-for-service and not research, and much of what you might consider fee-for-service may be considered research to them, then it would not count. It would just be subtracted out of the budget calculation completely as if it wasn't there. What is the typical impact cutoff star for funding? That varies. It really does. I would say in most cases where things in the well, I'd rather not even say because we've funded even stuff I was going to say. It really depends on the scores. If you're in the excellent range or near that, then you were probably at least up for consideration after our due diligence, but a lot of that depends on what exactly the summary statement says. So, you could be at 40 and the summary statement could sound really good, or you could be at 40 and the summary statement sounds really bad. and those two would, be very different in terms of consideration. What funding sources are available to move project progressively from screening to medicinal chemistry to preclinical models and IND application? I think I covered that during the course of this lecture. So if you're just running a screen in Ignite, if you've already screened and got compounds and you've got some data, Ignite can get you some preliminary data, or you could potentially come directly to Blueprint Air Therapeutics. The requirements for a chemical drug candidate to enroll in BPN. Well, besides applying through the cooperative agreement mechanism, the requirements vary according to those milestones that I presented today on where you're coming in from an entry point of view. The BPN contract, they're set. So the list that I showed you is the list that we have of vendors. And as I mentioned, if there are more than one vendor, it's going to be bid between them for your particular project. I can't tell you a priori which one's going to get it. What besides funding variability determines the choice between the two for a small business? Well, as I mentioned, you have to, in order for a small business to come to the U44, you have to meet all the SBIR rules. So ownership rules, can you keep all the, do you have enough money going internally to meet the requirements of 66% staying internally? Can you do all of that? If the answer to all that, you meet all the SBIR rules, then I would go under the SBIR. If you can't meet all those rules, then come under the UG3, UH3. That's really the biggest difference. The advantage to the U44 is for small businesses, you can charge a fee, and which you can't do under the UH3, UG3, UH3 methods. For SBIR U44, do we need to submit a commercialization plan as part of the Phase 2 grant? It needs to be part of this grant that you're applying. It's a fast track grant. You can't submit just the Phase 1 preparatory phase. You must. It's a fast track, and so Phase 1, Phase 2 from the get-go. Can we include a budget for preclinical efficacy studies and be responsible for conducting it through our collaborators for a lead that we aim to optimize? I believe again, if I'm not 100% on these questions, you can always ask me the question again via email, but I believe you're asking, can you budget for efficacy studies that you're going to use through a collaborator? And yes, it's just like any other NIH program. You can have the grant through you can have an investigator or another institution running an animal model for you. You have another institution running potentially the primary assay, and it can be a big collaboration. And that collaboration can include small businesses with academic institutions, small businesses with CROs, or academics with small businesses and vice versa and CROs. All of that works. There is not an STTR version, however, so just the SBIR version. Do you also deal with drug delivery vehicles and nanoparticles with bound drugs? This comes up, and it's a bit of a case by case. Typically, no, because this program is mainly driven towards small molecule drug discovery and development, and we don't have the contracts or the resources that really augment what you do. But there are some other mechanisms within NINDS and other institutes, both in the BRAIN initiative and other device initiatives that might be better for that particular application. We can talk more about that, but either myself or Nick Langhals in the device side could have a conversation with you about nanoparticles with your bound drug. But if you've already got a drug already optimizing and the focus is really binding to the nanoparticles, that's probably going to fall under that category. Would SBIR still go through the SEP and not the SBIR review panel? Yes, it goes through the SEP. They're side-by-side reviewed together. Did I hear you that contractors can claim IP? What you heard me say was inventors are determined by inventorship law in the United States, but intellectual property, we aim, for instance, the MedChem and the DMFP contracts have special clauses where they assign the intellectual property to your institution. the consultants will sign intellectual properties with you, again, to assign the intellectual property to your institution. I will expect if you have a collaboration that exists between you and multiple other collaborators institutions, that you will be able to produce a intellectual property agreement that says who has intellectual property, who's responsible for filing it. Because I want the grant to be with whoever's responsible and can make decisions on intellectual property licensing at the end of the day. If you license your program away, that does create a bit of an issue at times, and we need to discuss how that's going to work and make sure that we have handled it in multiple different ways. What is the relationship between the IGNITE and the BPN program? The IGNITE program is really a feeder program for the Blueprint. It allows you to get the preliminary data you need to have a strong BPN application or a strong CREATE if you have a biologist instead. It allows you to get your assay run, your screening assay run to identify compounds. It allows you to develop that compound, do some histochemistry, and get some in vivo data if you don't have it. Can the grant money be used for outside CRO for preclinical testing? Yes, it can be. So in the grant application, milestones that we will use BPN contractors, the milestone we use, how much detail should we provide? You don't need to provide very much detail. My instructions to the reviewers is, the assumption is that those are industry standard. So for instance, if you're going to use our BPN contractors for GLP tox studies, you really just need to say I'm running GLP tox studies, and you can say I'm going to, if you want to be complete, I'm going to run, repeat dosing in rodent and large animal, I'm going to run safety pharmacology, I'm going to run genotox and herd. And you pretty much have covered it through the BPN CROs under GLP conditions, suitable for filing my IND. Can we have the SOW included as we determine fit? Can we have the SOW included as we determine fit so the workflow is complete? I'm not sure what that means. So if you could restate that question a little clearer, I would appreciate it. To be clear, I'm going myself up to MS4. I wish I had these memorized. All right. So if you're coming MS4, they're saying you're coming, and I want the contract for MS5 and 6. So you're saying I've got a development candidate. So you're entering at the development, quote, the development phase, meaning I'm done with medicinal chemistry, and you want to do large animal DRF, you want to do cell screen polymorph work, tractable synthesis, all that good stuff. That is fine. You would say I'm a development project, but I'm going to do these pre-development activities as part of my preparatory phase. And my go, no-go decision is going to be a therapeutic index in a large animal DRF in order to move forward, and a reasonably tractable synthetic route that shows that I can make this in an economically feasible fashion. Not a problem. To do that, and then back to my company and subcontractor. That part I'm not quite sure about what you mean, but if you want to contact me and talk about more in detail, I'm happy to do so. But we can work with you on putting it back and forth. You can maintain your budget, maintain all the rules for if you're coming at SBIR. We're happy to be flexible. And in fact, one of our best projects actually, in terms of timelines, did it sort of a three-tiered approach where they used stuff themselves, BPN contractors, and then they also had outside funding to augment what they were doing so that while they could do certain things at risk that BPN required milestones to be in order to be met before we could start, they were willing to do some stuff at risk themselves to shorten their own timelines, which worked for them. If we use BPN for SAD trials, do we have to file human safety information with the application and don't designate this as a clinical trial grant? So I have to be clear about this one, because if you're coming in just for the clinical trial, you don't need the toxicology data, meaning you've already run your GLP tox study. You would not be in scope of this. You would be looking for the exploratory clinical trials, FOA, and not this program. If you're coming in saying, I've gotten through my pre-development and I'm really at the development phase, I need to run have my pre-IND, meaning I need to run manufacture my materials, run the GLP tox studies, then that's going to be something that we could do within the program. The deadline for the next grant cycle is August 9th, followed by February 11th.
Okay, right now we're at the end of the questions. I'll give you a chance to type in any last questions you might have. We'll give it like 1 or 2 minutes. If we don't,
a chance to actually drink some water.
Meanwhile, Dr. Cywin will drink some water.
I'm happy to answer any questions. And by the way, if you have additional questions or questions you don't feel comfortable putting forth here in a more public setting, reach out to me by email. Just so you know, Tim manages my calendar. If you're requesting a phone call from me, you can CC Tim on that request, and you'll avoid me having to send it to Tim to make that request. But the bottom line is he sets up the phone calls for me, and if I can answer your question by email, I'm happy to do that. I can usually do that the same day. Phone line calls with me can take a week or two sometimes.
And just to reiterate what we what we said, we will be sending out this presentation to everyone who is registered. So if you just dialed in and you didn't actually go onto the WebEx to register, please reach out to me so I can get your email so I can send everything. The idea is that we will be posting this online, but anytime we do, anytime with these recordings, sometimes they can go a little haywire. But the idea is that we will post this to our website. Someone who just, Mark, I see your question. Looks like you just missed it. The next application due date is August 9th, followed by the one after that is February 11th, 2020. All right, Chuck, we got another.
I always advise submitting a letter of intent. The purpose of a letter of intent, it's not required, but what the letter of intent does is allows the scientific review officer time to recruit people with the right expertise to review your application. That's his primary purpose. So I share those letters of intent with the scientific review officer, and so months in advance he can start to get the person with the right disease expertise in to review your application appropriately. Oops, what was that? I saw something there. I inadvertently did something. Go back to the question. Something to the presenter only. Give me a second.
No, you answered the question.
I did?
Yeah.
All right. Anything else?
Yeah. This was if BPN is running.
Okay. So if BPN is running clinical trial for our project, will the teams have inputs in the study design, clinical sites, and patient population, etc.? Yes. Our two clinical vendors right now are PPD, Austin, Texas, and Duke University in Durham, North Carolina. What we would typically do in a scenario where you have a clinical trial, and we typically fund the first-in-human SAD studies, is get a protocol synopsis and bid that out to the two vendors. We are looking, the program doesn't have unlimited funds, and the designs that we typically do for our first-in-human studies are simple SAD studies with the usual number of patients per cohort, like typically 6 and 2 placebo, and we might run like 6 cohorts. They're usually healthy volunteer studies, but there could be cases where depending on your indication, that might be different. But most cases, it's going to be considered healthy volunteer studies unless there's a reason not to. Keep in mind that the FOA says things focused on beyond phase 1 are out of scope. And so presently, we're not planning on doing any phase 1-2 combined studies that we would do. But again, if you have specific questions, I'm happy to talk to you about those. Again, I answered the question of how many awarded per year. It really depends on the number of applications I receive each round and their scores. It varies. 2 to 4 is a typical year. We presently have 11 active programs running simultaneously right now, with several going to the clinic. So, all right. Is the BPN website down? Yes, I think it is. It wasn't earlier today, but I went to go right before this webinar, and it gave me an error too. So it may well be down. I don't know what the issue was. I didn't have a chance to inquire before I came upstairs, but I actually went to look at the website right before I walked up here to the conference room to do this webinar, and I too could not get to it. It was working fine yesterday, so I would just check again shortly. It should be fixed. The question that was up there.
Yeah.
We have 2 efficacious compounds that both suffer from PK stability issues. Can we use to formulate the compound to get a better PK profile? Then use the new formulation that's got IND enabling. Yes, you could do that. You would have to, that would be a development project for which you're really focused on formulation. I would say if you're going for an oral formulation, I would consider doing that, contracting that as part of what you do, so that you could get the right expertise for your particular problem, because that's a really particular issue when you're trying to improve the exposure of a compound that or PK. And I assume by PK you really mean exposure and not the stuff is metabolically unstable. And because if it's not, it's really metabolically unstable, and that's not stability of the compound itself, then I would question whether you have the right compound and whether you really should make better compounds first as your goal. Not knowing your SAR, not knowing what you did, you may well have gotten to the point where that's just not feasible. I see someone put, Beth, you put in a number of questions that don't seem to have made it to me. You can certainly ask questions after the webinar to me directly. Or you can try sending in wherever you just sent in your response came through just fine. I'm not sure where, I don't, Tim is scrolling right now. I don't see any other questions from you. So if you want to resubmit the questions, by all means you can now or after the seminar. I'm happy to try to answer them as quickly as I can. There is a program complementary to the BPN for large molecules called CREATE. It's not completely complementary because it's only the NINDS program. But if you're within NINDS's mission in the Neurology Institute, then the program called CREATE is complementary. Because of the vast differences between the different kinds of biologics, they don't have the suites of contracts we have. They have some limited consultants. So there you would really want to put together your team yourself and probably end up submitting an ARA to come in with the budget, but it's a very similar program, milestone driven. Are one's chance of getting even greater if you have in vivo data? I'll be honest and say if you have in vivo data on your compound series, chances are it's going to be stronger application. That being said, I've seen people make very successful applications for which they have data only on literature compounds, and they make a strong link for why this compound has intellectual property is going to be better than the literature compound, and without having that in vivo data. And I've seen where they have development projects where they have really strong in vivo data on the primary compound. They've got it down to the candidate compounds, and they don't have in vivo data on that yet, but they say, based on this profile, we expect when we recapitulate the data, it'll be good. That's going to be our go, no-go decision point to move forward as part of their preparatory phase. Is Ignite open to small businesses that would qualify for SBIR grants? Ignite is open to small businesses, and I would have to check to see if I don't believe there's a SBIR specific version presently for Ignite. I would ask Becky Roof about that to be sure, but I know the FOAs I put up today are available online, and I'm about 95% sure that take both businesses and academics, foreign components, and foreign institutions.
All right, with that, I think we're at the end of our Q&A webinar. Thank you to everyone for joining.
Yes, I agree. Thank you all for your attention today. Happy to answer questions. I do have, I'll be completely honest, I have limited availability over the next couple of months between this program and the HEAL optimization.